Good morning, students. Today we will continue with the next topic, that is in the same topic, pure cultures. Yesterday's class we have seen what are pure cultures. Pure cultures means these are the cultures which we are getting, which are nothing but they are of only one kind of microorganisms. That means the culture consisting of only one type of microorganisms or one kind of microorganisms is called pure culture. How to isolate it is yesterday's class we have seen, that is in the previous class we have seen, that is serial dilution technique, streak plate method, spread plate method, pour plate method, next is your enrichment technique, next is special culture method. All these methods, enrichment method and special culture method, all these 6 methods we have seen for the isolation of pure cultures. And next, for maintenance of pure cultures, what we have to do for isolation we have done. Now coming to the next one, the characteristics of pure cultures. See here, the characteristics of pure cultures are, these pure cultures, they can be identified. After isolation, you have to identify whether they are actually pure cultures or not. For that identification, what you have to do is, see here, see here, for its identification what you have to do is, simply you have to go for morphology. Morphological identification. What are the identifications, morphological identifications? What is the meaning of actually morphology? Means external features. Looking at the external features, you have to identify it. For that, you have to see the shape, size, arrangement, okay, arrangement. Next, coming to the next one is movement, flagella, these are the 6 generally observed characteristics of morphological characteristics. Coming to the shape, whether the bacterium is cocci, that is spherical, or they are bacillus, that is rod shaped, or they are spirillum, or they are vibrio shaped. That means you have to see the shape of the organism. Take a small amount of your bacterial culture on a slide and observe it, microscopic slide, and observe it under a microscope, so that you will come to know the morphological characters of your bacterium, whether they are in the bacterium is showing what is its shape, circular or rod shaped or spirillum or comma shaped, whatever it may be, these are the shapes of your bacteria. You have to identify it, that is shape. Coming to the size, you have to measure the size of the bacterium with the help of an instrument called as micrometry. That is, metry means meter, micro means small meter, which is useful in measuring the length of the microorganisms. So with the help of, you are not taking a scale and you are not measuring it, simply the scale will be placed in the ocular lens, that is eyepiece of the microscope, from which you are viewing the microscope. అంటే మైక్రోస్కోప్ని చూసే లెన్స్ లోపల పైనుంచి ఆ పై లెన్స్ ఓపెన్ చేసి దానిలో ఒక చిన్న స్కేల్ లాంటి స్లైడ్ వేస్తాము. ఆ స్కేల్ వేయగానే ఆ లెన్స్ అది క్లోజ్ చేసేసి పెట్టగానే ఇక్కడ మైక్రోస్కోప్ స్లైడ్ మీద పెట్టి స్టేజ్ మీద పెట్టి మనం చూస్తుంటే, మైక్రోస్కోప్ స్టేజ్ మీద పెట్టి చూస్తుంటే పైనుంచి ఆ స్కేల్తోని మనం మెజర్ చేయొచ్చు. దట్ ఎక్స్పెరిమెంట్ యూ హ్యావ్ ఇన్ యువర్ ప్రాక్టికల్స్, దట్ విల్ బి డూయింగ్ and you will know how to measure the microorganism. So with the help of micrometry, you can measure the size of the microorganism in micrometers, because they are very small in size, that are not visible to our with our naked eyes. So for that reason, the size of the microorganism is measured in micrometers, and its measurement is made with the help of micrometer, and its measure is measured with micrometer units, micrometers. Now coming to the arrangement, whether this is the shape. Suppose bacteria is there, it is single celled, or the cells are joined, double cells, or the cells are joined like this, a chain, staphylococcus, or this is unicoccus, monococcus, diplococcus, staphylococcus, that is a chain of organisms are there. And sometimes the microorganisms are joined like this, just like a cube. In front I am drawing 3-4 circles, on back side also it consists of 4 circles just like a cube, and this is called sarcina. If it is bacillus, the arrangement is unibacillus, monobacillus, diplobacillus, a chain of microorganisms are present, that is staphylobacillus. Next, the microorganisms are joined like this, and backside also they are there, that is called as a sarcina. So the arrangement of the microorganisms can also be observed with the help of your micrometry or microscope. Okay. Now coming to the movement, whether the microorganism is motile or non-motile, you have to see whether it can move or cannot move. If it can move, they are called motile microorganisms. If they cannot move, they are called as non-motile microorganisms. Now if they can move, then what is this? If they cannot move, there is no need of flagellum, but if they move, then with which organ or appendage they are moving, what is the organ that helps in the locomotion or movement of the microorganism. For that we will go for flagella. The flagella is a structure which is present like this, which is a shaft like structure which helps in the movement of the microorganism. So for that, the flagellum, whether the flagella are, see here, monotrichous, lophotrichous, next amphitrichous, next peritrichous. So these are the 4 types of arrangement of flagella. Monotrichous means this is only one side flagellum is present, then it is known as monotrichous flagellum. Next, lophotrichous means, see here, a bunch of flagella are present only on one side of the bacterium, then it is called as lophotrichous. Coming to the amphitrichous, the name itself indicates that wherever you get amphi means both. Why? Because you remember frog. All of you know that frog is an amphibian. So frog can live both on land and water, on land and in water. So for that reason, as frog can live both on land and in water, that means in both areas it can live, so it is known as amphibian. So here amphitrichous, wherever you, ambidextrous means who can write with both the hands. Amphi means both, dextron means hands. So ambidextrous, that is an English word, linguistic word. Here amphitrichous, why we are using trichon? Because the other name for flagellum is trichon. For that reason it is known as amphitrichous. So a bunch of flagellum are present on both sides, just like it looks like a eclair's chocolate. It just looks like a chocolate. Now coming to the peritrichous. Peritrichous means this is the bacterium. The flagellum is present throughout its body. Okay, the flagellum is present throughout its body. This is known as a peritrichous flagellum. Okay. Now coming to the capsule. Let us see what are spores. Spores means these are the reproductive organs. When they fall on the ground, they will start germination, just like your fungal organisms. Here some of the microorganisms, they produce spores. So whether they bear spores or they won't bear the spores, that is based on the identification of the spores. Next, coming to the capsule, whether the bacteria, suppose this is your bacterial cell, whether the bacterial cell is surrounded by this is your cell wall, this is your plasma membrane, and this is your capsule. So some bacteria consist of capsule and some do not have capsules. Generally the capsulated bacteria, they are pathogenic. That means they can cause diseases, because if you use medicines also, due to the presence of that thick capsule, the microorganism is protected from the effect of your medicines. For that reason, the microorganisms consisting of capsule, they are generally pathogenic, that has the capability to produce disease. Okay, this is about the morphological characters: shape, size, arrangement, movement, flagella, capsule and spores. Next, coming to the next one, elevation. See here, next one is elevation, second one is margin, third one is shape of margin. See here, slowly I will tell you. Next, shape of colony. See here, coming to the next one, morphological characters only. Coming to the shape of the margin, first we will see shape of the colony. Whether the bacterial colony, suppose you are inoculating the bacterial cells in a plate and the bacterium is producing colonies. Each and every colony consists of thousands of bacterial cells. So ఒక్కొక్క కాలనీలోనూ thousands of bacterial cells ఉంటాయి. ఆ బ్యాక్టీరియల్ సెల్స్ అన్నీ కలిపి ఉండే ఆ ప్లేస్ని ఏమంటారంటే దిస్ ఈజ్ కాల్డ్ బ్యాక్టీరియల్ కాలనీ. నౌ సీ హియర్, when you are talking about the bacterial colonies, what is the shape of the bacterial colony? I have drawn circular shape, the spherical shaped colony. Some colonies, they will be like this, irregular colonies. The colony will be irregular. Sometimes the colony will be ellipsoidal. Next, the colony will be rhizoidal. That is just like a root like. The colony will be rhizoidal. That means, see here, the bacterial colonies, they will be having different shapes: spherical, elliptical, spherical, ellipsoidal, irregular and rhizoidal. These are the different shapes of the colonies. Now, coming to the next one, texture of the, see here, coming to the margin of the colony. See here, the margin, this round thing is known as margin. Coming to the margin of the colony, the margin of the colony, it may be smooth or it may be wavy. See here, it may be wavy or it may be with irregular shape. The colony margins may be smooth, wavy or irregular. Now, coming to the shape of the margin, this margin may be, it may be smooth or rough. Okay, the margin may be smooth or rough. If it is wavy also, if it is smooth. If it is rough means, see here, here and here something rough structures will be there. This is known as a rough margin shape. This is the smooth margin shape and this is the rough margin shapes. That means, when you look at the margins, the margin is either circular or wavy or irregular. Coming to the shape of the margin may be smooth or it may be rough. Okay. Now, coming to the elevation. Elevation means, the elevation is flat or concave, convex. See here, concave, convex. See here, embossed. అర్థమైందమ్మా? చూడండి, the elevation, suppose this is your bacterial culture. Bacterial culture is growing. Bacterial culture, if it is growing flat on the surface, that is called as a flat elevation. If it is growing just like dip like, on the medium it is producing a dip like structure and they are growing, then that is called as a concave depression, concave elevation. Some will be growing convex, just a top. Next, some will be showing embossed. What is the meaning of embossed? see here, when you look at some pictures they will be giving a 3D type of image, so that they will be shown just suppose this is the surface, on that surface it will be seen here, that means embossing. What you can say is, see here, when you are having a dress, on that dress you are doing patchwork, that is bottom pajama is there, pajama's cloth we are taking a little bit and they will be placed here and there on your surface of your fine kurta. On that kurta if you are placing it and stitching it, that is the bottom pieces are embossed on our kurta. That means pajama cloth is embossed on your kurta. Same as that, here some of the bacteria will be looking just on the top of your that one. This is known as embossed elevation. Now coming to the texture. If you touch it, how it will be? The texture will be fabric, that is it is just like your cloth, or it is shiny, or sorry, it is fabric or leathery. It may be fabric in nature or it may be leathery in nature. Next, sixth one. When you look at how it will be, it will be glassy, that is it is transparent, it is looking just like glass like, or it is transparent or opaque. It is giving the appearance of glassy, shining glossy, or transparent or opaque in nature. It is glassy or shiny and transparent and opaque in nature. That means by looking at the surface only you can give these many. See here, 7 plus 6 means 13 to 14 characteristics. By looking at the organism you can identify it without going into the organism, simply by observing the structures. Now coming to the next one, when you talk about the staining procedures, see here, by giving a type of stain, how you can identify the bacteria. When you are identifying the bacteria with the help of stain, with the help of staining, when you are identify the bacteria, now see here, this bacterium, when you are staining with bacteria, if you go for simple stain, differential stain, next Grams staining. These are the three methods of staining. Simple staining already you have done in your first semester. I made you to do this. I asked you to bring a slide and I made you to perform this experiment and observe the shape of the microorganism. So simple staining is a method. In this you will be using only one color agent, one dye. Next one, this simple staining helps in identification of the morphology. Morphology of bacteria. What is morphology of bacteria? Just now what we have seen, that is whether it is spherical shaped, rod shaped, spiral shaped or vibrio shaped, whatever it may be, comma shaped, the shape of the bacterium can be identified with the help of simple staining. Coming to the differential staining. This differential staining helps in staining each and every organ differently, that is flagellum. Flagellum, for the identification of flagellum you can go for differential staining. Cell wall, capsule, next pili. Like that, whatever organ you want to identify and observe, then go for differential staining. Each and every organ will be stained with different colors and you can easily observe or study each and every organ separately, clearly, equally. Now coming to the Gram staining. Gram staining is a type of staining. In this you will be going for, see here, in this Gram staining you will be using two dyes, that is two coloring agents or two dyes, and this Gram staining helps you to go for identify the gram positive and gram negative bacteria. Both the bacteria, that is the gram positive and gram negative bacteria, can be identified with the help of gram staining. Okay. So this is your staining process. Now for the identification of the bacteria, whether they are resistant, next one is resistance, next one is metabolism. See here, coming to the resistance and metabolism. Resistance means whether the bacteria is resistant to high cold or high very low temperatures, high hot or high temperatures or low temperatures, that is very heat or very cold climates, whether it can, the bacterium can be resistant, is the bacterium resistant to high cold and high low temperatures, high heat and high low temperatures, that is high heat and very cold temperatures, whether it can be resistant to this can be identified, and whether the microorganism is resistant to disinfectants, and Lysol, Harpic, all these disinfectants, Nimile, all these disinfectants we are using to clean the floor, Dettol, Savlon, all these disinfectants. When you are using these disinfectants, whether our bacterium is resistant to these disinfectants or not, you can study it. Coming to the metabolism, whether all the enzymes are produced or not in the enzyme, in the microorganism to undergo metabolism like excretion, reproduction, digestion, etc., replication of DNA, etc. Next is whether they can produce hormones or not for protein synthesis, enzymes are produced for mRNA synthesis, DNA synthesis, mRNA synthesis, DNA synthesis, RNA synthesis and protein synthesis, all the enzymes are synthesized or not, all the hormones are synthesized or not, whether the bacterium require oxygen or whether they are can grow only in the absence of oxygen, whether they are aerobic, in other words whether they are aerobic or anaerobic, everything can be studied with the help of metabolism. Now we have seen all these things. Now coming to the staining procedures, that is not staining, you can go for some of the tests for the identification of bacteria. The tests for the identification of bacteria are, a few tests are there with which you can identify the bacteria very easily. What are they means? They are acid production producing bacteria, indole producing bacteria, methyl red test. Next, see here. The first test is a sugar fermentation test. In this sugar fermentation test, you will be identifying during fermentation acid is produced. Now, during sugar fermentation, whether this is also known as acid producing bacteria. That means identification of acid producing bacteria. For this, see here, what you have to do is take your bacterial cells. See here, take your bacterial culture or take your culture medium plus add phenyl red. Leave it at 48 hours, at 37 degrees centigrade, add your bacteria. Take culture medium, add a little 1 or 2 drops of phenyl red indicator, then add your bacterial culture, fresh bacterial culture that is grown 24 hours back, and place this bacterial, incubate this bacterial culture at 48 hours at 37 degrees centigrade, for 48 hours at 37 degrees centigrade. Next what happens is, the bacteria will grow and this medium turns pink. If the medium turns pink, it is an indication that this organism, that is the bacteria, it can produce acid. It helps in the production of acid. Along with that, it also produces gases. That means if the medium, after growth of 48 hours of growth of bacteria at 37 degrees centigrade, if you observe the tube, culture tube or culture vessel, you can say that the medium has converted into pink color. That is an indication that This, it is sugar fermentation has taken place, because in your culture medium sucrose or glucose is there, that glucose fermentation has taken place and acid has been produced, along with some gases are also produced, because the medium has converted into pink. Without going for any test, you can say that if the medium has converted into pink color, the bacterium is helpful in sugar fermentation, acid production and gas production. This is identification of acid producing bacteria test. Second one is here, next test is your Imvic test, or this can also be called as indole production. This can also be called as indole production test, or identification of indole producing bacteria. Okay. Now see here, in this test what you will be doing is take bacterial culture. Okay. Take bacterial culture, and in this bacterial culture you grow it at 48 hours at 37 degrees centigrade. Take some bacterial culture, grow it at this grow it for 48 hours at 37 degrees centigrade. Next, after that, to the bacterial culture you add 0.5 ml of Cavox reagent. Shake it. Shake it well and keep it aside without disturbing it for few minutes. If it is showing pink color, see here, if the bacterial culture has retained, it has taken up pink color, or it is showing pink color, then that is an identification that indole is produced. That means in the bacterial culture you will be adding tryptophan medium, or you can call it as a trypton medium. See here, you can call it as a tryptophan or a trypton is taken in your bacterial culture and incubate it at 48 hours, incubate it at 37 degrees centigrade for 48 hours. After that you take the culture and add 0.5 ml of Cavox reagent, shake it rigorously or shake it well and keep it aside, so that shake it rigorously and keep it aside. If the medium has converted, bacterial culture has converted, the bacterial culture medium has been converted into pink color, if it is showing pink color, if the bacterial culture medium is showing pink color, that is an indication that indole is produced, because already the precursor of indole we have used, that is tryptophan. That means in your MSc entrance you will be asked, the IAA, that is indole-acetic acid, is produced from a precursor. What is that precursor? It is a tryptophan. Okay. Tryptophan is the precursor for the production of indole-acetic acid. This is an MSc entrance question. So in your PG entrance you can expect this question, that is the precursor of indole-acetic acid production is tryptophan. Okay. Now coming to this third test. The third test is, see here, in this third test we are going for methyl red test, or you can call it as a MR test or methyl red test. In this you will be identifying, or you will be checking the efficiency or efficacy or efficiency of your old cultures. Suppose old culture is there. Till now I am talking about the fresh culture, fresh bacterial culture. Now suppose old culture is there, that means in our lab we have bacterial culture, and that bacterial culture we kept it in your lab, refrigerator, and during summer vacation 2 months we are not coming to college. After coming to college, for 1 month we will be taking the classes, we are busy with the classes, and after 1 month your practicals are starting. So when your practicals are about to start, myself and Kamlakar will go and check for the old cultures, whether they are efficient or not. Then what we will do is, we will take these old cultures and whether they can grow at 4.5 degrees centigrade, or 4.5 pH. That means whether they can grow or they are efficient. If you do experiments with these old cultures, it is okay, you will get, will you get the results or not, we have to go for checking of the old cultures. Take the old cultures, whether it can produce acid, or whether they can grow at a pH of 4.5, what we have to do is take these old cultures, to them we will be adding methyl red. When I add methyl red and keep it aside for few minutes, for 5 to 10 minutes, then it will be showing two types. One is red, yellow. There is a chance of getting two colors. One is a red color, the other one is a yellow color. If you get red color, it is positive result. If you get yellow color, it is a negative result. What is positive and negative? It is not COVID. It is only checking the old cultures. So, if you get a positive result, that is red color, that means our culture is efficient and its efficacy is okay. You can take these old cultures and use for your experimentation. If it get yellow, it is of waste, you have to throw that culture. That means if you get red, it is positive and you have to go for your experimentation. That is okay. Next is last test, that is citrate test. Citrate utilization test. What is citrate utilization test? See here. In the citrate utilization test, you will be taking citrate as a carbon source. Okay. The citrate is taken as a carbon source in the culture medium. Now, in this you will be inoculating bacteria and leaving it aside. Slowly the medium, suppose this is your citrate and you have inoculated bacteria. Now, at the end of your experiment, here the medium is there and the medium is completely filled with bacteria and the medium becomes turbid. The turbidity indicates that the citrate is utilized as a carbon source and the bacteria have grown. This test is known as a citrate utilization test. Can you see the difference between these two? Here the medium is transparent, only 2 or 3 microorganisms are there, but after 37 degrees centigrade for 48 hours, the bacterium will grow in numbers, multiply, so that the number of microorganisms will be increased. The bacterial culture medium, here it is transparent and it becomes here turbid. The turbidity tells us that the bacteria is utilizing citrate as a carbon source. So, this is the fourth test of your pure cultures. So, with this we have completed the characteristic features of or characteristics of a pure culture we have completed. In the next class we are going to see the sterilization techniques. Okay, thank you students.